My name is Laura Edwards, and I'm with South Dakota State University Extension, and I will be one of the moderators, I guess, for today's presentations. We have two great speakers here today for this fourth webinar in our webinar series. And just a reminder, this is a Climate, Ag, and Water webinar series for Extension professionals. And this is fourth of 6 events that we're holding. And we do have a website where these will be archived, and so you can see them later, or your colleagues can see them later on the climateagwater.wordpress.com, and we'll share that website again with you later. That's been funded by North Central Region Water Network, have been Hans Schmitz at Purdue, Peter Tomlinson at Kansas State, and also Tyler Williams at University of Nebraska. And Dennis Toddy at South Dakota has also been involved in this project. So thank you all for attending today. I'm looking forward to two great presentations with Matt and Megan. At this time then, I'd like to introduce Matt Reeves. And Matt is a research ecologist with the Rocky Mountain Research Station in Missoula. He has degrees in range management, environmental resources, and his PhD is in remote sensing and ecological modeling of rangeland and agricultural environments, received in 2004. He specializes in applying remote sensing and modeling to characterizing ecological dynamics of rangelands. He's also the co-director of the Northern Plains Regional Climate Hub, the new USDA Regional Climate Hub up here. And his research follows 4 basic themes, including climate change, vegetation dynamics, anthropogenic disturbance, and decision support tools for improving management efficacy. So with that, I'll turn over to Matt, and I believe his title is Projections Suggest Uncertain and Possibly Perilous Future for U.S. Cattle Operations. And
good,
Matt,
well thanks.
Yeah, you're ready to go.
Yeah, thanks Laura. I wanted to start with a question to make sure you could hear me.
Yeah, we can hear you just fine.
All right, well thanks everyone for joining today. I look forward to talking with you about this subject, which represents 3 or 4 years worth of snooping and investigation with 2 colleagues, Karen Bagney, who is a cooperator with the Forest Service, and Dr. John Tanaka, who is Associate Director, Wyoming Agriculture Experiment Station, among other things. He wears a variety of hats. I guess I don't need to spend a lot of time talking about why someone may be interested in the aspects of climate change that might influence cattle production in the United States going forward. We don't need to look very far from headlines of various periodicals to understand that things are changing. And we wanted to understand perhaps how things might change in the future with respect to the AR4 climatological scenarios. And we did this, as you'll see, using a variety of biophysical indicators that tell us something about the suitability of habitats, if you will, for cattle operations going forward. So we really wanted to look at the vulnerability of livestock production to climate change and identify regions that may be most at risk for reduced capacity, and then identify also what is the most likely mechanism that would be responsible for reducing the capacity. And to do that, we looked at 4 basic elements, or 4 biophysical indicators, if you will. Forage quantity being one, forage dependability, so the interannual variability of forage, of course, is a big determinant of risk in some cases. Heat stress index, and then also vegetation type shift. And with that, you can imagine that as climates change, as the periodicity of precipitation and temperature and these types of things move and reordinate in the future, you can imagine the possibility for different vegetation types expressed across the landscape. And then we talked about what the study was comprised of, which is understanding the vulnerability of livestock production to climate change. And I was just introducing the 4 biophysical elements when Laura told me that you couldn't see my slides. So here we are. They are forage quantity, forage dependability, heat stress index, and vegetation type shift. And that was under 3 climate change scenarios of AR4 ilk, A2, A1B, and B2 for those of you that are into that language, 2001 to 2100. So the idea was to calibrate against present day conditions of vegetation type, quantity, forage dependability, heat stress, and then compare those elements in the future to present day. So I'm going to set the tone briefly with one slide with respect to the climate that we are examining vulnerability across. And in simple terms, the panel A represents U.S. precipitation and how it's projected to change in the future. And you see that across these different scenarios, which are represented by different colored lines, there's really no trend overall in the total amount of precipitation with respect to these changes in precip. What we do see, though, is in panel C, we see that obviously regional differences come out pretty clearly. And the two biggest things to take home here is that it looks like things get perhaps slightly drier, plus or minus, in the southern United States, the southern extent of our study area, and perhaps a little wetter to the north across all 3 scenarios, looking at them in concert. With panel B, which is temperature, things are different. You see a general warming trend overall with respect to the temperature. Panel B shows us something about the regional differences. I want to point out that the cool tones here, the greens and the blues, don't mean that things get cooler. It just means that they warm at a slower rate than the warmer tones. And that's an important feature of this, I think. So talking about those climate effects on the 4 biophysical indicators, first we wanted to look at forage quantity. So we used a biogeochemical cycling model to forecast what the rangeland production might look like in the future. And of course that takes into account carbon dioxide enrichment through time, looks at carbon and nitrogen dynamics, disturbance. Of course, climate is a big driver, that being temperature, precipitation, solar radiation, and these things. And we need a starting point. So we need to know roughly present day species composition, especially with respect to cool season and warm season mixes of species. This work was published in 2014 in Climatic Change. With respect to forage dependability, you'll note that I did mention in the beginning we're comparing future trends with present day trends. So what I wanted to set the tone was this map represents over the last 15 years the variability of forage, if you will, across the landscape, and cooler tones represent areas that have experienced higher variability. The warmer tones are areas where things have not changed as much on a percentage of the mean basis. So this is coefficient of variation here. With respect to vegetation type shift, data that were used to represent this biophysical element was the MC2 model, which is a dynamic global vegetation model. And what we wanted to get out of this was, in general, are the landscapes responding to climate by getting woodier, or are they getting more herbaceous in nature? And the MC2, of course, has been published in a variety of venues, but it enables kind of an assessment of we superimpose climate, we superimpose fire regimes, and we begin to tease out what that might mean for species assemblages in the future. With regards to heat stress, again, another one of the biophysical elements we examined, we used a well-known equation that's shown up here from Brown and Brandle et al. In Livestock Science. And basically what we wanted to see was how this changes in time. It's very sensitive to temperature, and it's also very sensitive to humidity. So increases in either of those variables will change the heat stress index. And the way that we took all this information and combined it to understand the combined effects of these biophysical indicators on potential livestock production, we looked at the change relative to present day, and that being the contemporary baseline, if you will. And then we categorized these changes so that they were all on a common baseline, if you will. In other words, you can't compare a change in heat stress index to a change in forage without some kind of translator between the two. And so we looked at percentage changes in these variables through time and assigned classes, -2, -1, a 0, a 1, and a 2. So again, we're looking at general changes and how these compare to one another in time. So here's, we'll go through a series of results and then end with a brief discussion of what I think it could mean. This is forage dependability, which basically, remember, that talks about how variable our forage is projected to be year to year. So interannual variability of forage, again, with respect to present day. So in these maps, the way you interpret all these outputs is they're all color-coded the same. Remember, there's 5 scores, a -2, a -1, a 0, a 1, and a 2. If I'm positive, I am greener, and that's good. That means my forage dependability increases. That means I get less variable through time in response to these changing climates. But if I'm yellow or red, that's bad. That means I'm increasing my variability year to year, and my ability to predict what's going to happen next year with forage is poorer, again, with respect to present day. And as you can see, all the scenarios are represented, all three, A1B, A2 and B2, and at 2 points in the future, 2060 and 2100. The two take-home messages are, by and large, overall, on average, most things tend to get less predictable through time. The exception is with the B2 scenario, which some people claim is just kind of a past argument that we wouldn't see this scenario take place in the landscape anyway. Note that in the north, things become less variable. In the south, they become more so. That's in general how I describe that situation. But what about forage quantity? Across the board, and again, the color-coded the same. Across the board, the number one take-home here is, doesn't matter which of these scenarios on balance you're looking at, they tend to project greener environments to the north and browner environments to the south. And of course, I'm paraphrasing and using finger air quotes here, but that's the general point. And there are 3 papers that have been published that substantiate this greening of the north and browning of the south. The first just came out. Productivity of North American grasslands is increased under future climate scenarios despite rising aridity. And that was a very interesting read. If you've not done so, I'd suggest you take a look at that. Our own publication, which suggested that, again, things tend to brown in the south, green in the north, and then finally the study by Morgan and others where the C4 grasses are prospering as carbon dioxide eliminates desiccation. What he found in the face experiments in some cases is production ranged anywhere from 10 to 20 to 30% increase under these carbon dioxide enriched environments with no change in precipitation plus or minus. So we're finding some evidence that's beginning to substantiate this idea of things becoming a bit more greener in the north and browner in the south. Okay, on to vegetation type change. The way we interpret these outputs is that if I'm green or if I am a neutral color and not red or yellow, so anything but red or yellow, that means I'm becoming more herbaceous. The shrubby component of the ecosystem is declining. And again, broken out by scenario and by 2 points in time, 2060 and 2100. Overall, there's kind of that east-west boundary sort of transition along what would be about the tallgrass prairie cutoff point you can see far to the east. This has to do with fire regimes increasing in that, supposedly increasing in that area, removing some shrubs in some cases. There's some other causes too, but overall that's the dominant effect, and of course elsewise, eastern Washington, Nevada, Utah, we see an increase in woody species regardless of the scenario overall. Heat stress is a very interesting component of the analysis. So you're looking at this and thinking, there's a giant swath of red and what happens? Well, remember we're comparing projections of heat stress index to present day. In the north, we don't experience a lot of it, so small changes mean a lot. And livestock operations are, in some cases, sort of calibrated to the local conditions under which they're behaving, or under which they're thriving, hopefully. And further to the south, they've had heat stress since they started raising cattle there for a long time. So heat stress has been a big player of those operations, and so the effect of adding a few more days of heat stress perhaps isn't as much of a shock to the system as it is to the north on a percentage basis. So when we take a look at these indicators across various regions and look at the relative vulnerabilities, there's a couple of things that jump out at you. And again, the rows represent the biophysical indicators. So in this case, starting with forage availability, which is the NPP, the veg, which is the vegetation type change, HSI, heat stress index, the VAR, which is the variability of forage. The black lines indicate the mean response across all the scenarios, so they're combined, it's an ensemble approach, and the gray bounds represent the deviation with respect to that mean. So A1, B2, B2 comes all together to represent the black lines, but the variation, or the difference between scenarios, is represented in the gray. And what we see is the northern Great Plains, interior west, to some degree the southern Great Plains, eastern prairies, all tend to experience increases in production of forage, although the northern Great Plains, I think, to a greater degree. The other thing to note would be that the heat stress index in all cases begins to have an immediate, rather punitive effect, I think, on our projections of habitat suitability, if you will, for cattle operations. The variability of forage is kind of all over the map, and that's also where we see the greatest amount of disagreement between the scenarios, is with respect to year-to-year variability in climate and how that impacts the forage. I'm going to skip this slide and just begin to talk about the map instead. What I want to do is point out a couple of things. The first is the upper two panels represent the amalgamation of the four elements, so they were added together. So in this case, if I'm -2 in all the categories, I'm a -8. Likewise, if I'm a +2 in all categories, I'm a +8. And you can see those colors represented in the middle of the screen. The -8s and the very high minus numbers are reds. The very low, or the very high numbers rather, that's positive overall, and they are represented by cooler tones. And the neutrals, of course, represented by grays or soft colors. You see a couple of things of note. First, there's that band that runs from Texas, Arizona, New Mexico, Nevada, California, Utah. There's that crescent, if you will, that comes out in many of the indicators, and of course here in the overall result, suggesting decreased performance, and in some cases marked decreased performance possible with respect to these four biophysical indicators of cattle production. What the bottom panels represent is the disagreement between the scenarios. In this case, cool tones mean, so the blues and the greens mean that the scenarios disagree on the end result. If I'm warmer tones in the bottom two panels, I agree more on a relative sense. So Texas, for example, wow, things look pretty bad from the vulnerability perspective, but the models really don't agree, and much of that has to do with precipitation. So that's the overall result. Again, greening in the north and in the northern Great Plains, north central Great Plains, but a browning in the south overall and a decrease in conditions. So southern rangelands become more vulnerable, Northern rangelands, it's sort of unclear, but trend towards positive. Relatively higher uncertainty in the southwest. But here's the linchpin for me. After doing some more snooping, we can see that recent trends suggest that our projections yield some insight. What do I mean by that? If we look at contemporary trends of satellite-derived vegetation performance, which is panel B, and then look at combined analyses of temperature and precipitation from, for example, PRISM, the PRISM climate data set, we see overall that this change in growing conditions, which is represented panel A, has here's that crescent again. So from 82 to 2012, we see this browning crescent, if you will, that I had pointed out in the projections. So growing conditions have gotten worse by my reckoning in that area of red, yellow, orange, whereas they've improved, and in some cases quite a bit, in the north central plains, also somewhat in eastern Montana. You can see that pretty strongly. If we look at just trends in vegetation, 2020, 2013 in this case, satellite-derived NDVI, these are trends. We see again Texas is a big, big loser, as is eastern New Mexico. Nevada, also a loser. Southern Arizona, also a loser. I wouldn't really trust the band that you see in North Dakota, the wide forked yellow, because there's a lot of interference that we need to continue to kind of clean up, interference, if you will, from the agricultural setting that may be quite pervasive there. Anyway, this is suggesting, again, growing conditions have gotten worse by my reckoning in the south, and this is reflected to some degree in the satellite-derived vegetation analysis. One more piece of evidence that's linked that may suggest that the trends we're projecting have some validity, or at least have some linkages to what we see with drought. So the bottom panel here, the colorful thing, is if we took all 52 weeks from the USDA Drought Monitor and summed them all together across 15 years, so it's a cumulative assessment of the USDA Drought Monitor, we see that the red spots are hot spots that indicate where cumulatively there has been more drought categories and a higher amount. And if you compare that with this crescent, I don't know if it's a crescent, but if you compare it, I guess it's more like a bat. But if you compare it to the bat in the upper two slides, which represent our index, there's an interesting, I think, regional kind of correlation here. Again, the upper panel suggesting reddish, brownish, yellowish colors. That's our cattle vulnerability index. And the thing to the south is where we've been seeing drought. Recognizing the top panels represent projections, the bottom panel represents somewhat of a contemporary analysis. And so I mentioned that the recent trends suggest some projections that may yield some insight. You know, we're seeing some things unfolding today that look like they intensify in the future. Although we acknowledge this is just sort of one method for evaluating impacts, and we didn't take into account adaptation strategies such as switching breeds, which is a very interesting topic that we'll be hearing about very soon, and also switching, maybe switching to other livestock systems altogether. So we haven't talked at all or factored in adaptations. Only the potential impacts based on these three scenarios into the future compared with a contemporary baseline. So with that, I hope I've left some time for questions, and I was glad to be able to share this with you today. So thank you for this opportunity.
All right, great. Thanks, Matt. If there are any questions for Matt directly, we'll take those at this time in the chat box or the Q&A. Hans, was there a question there in the Q&A?
There is. Shannon would like to know if you could provide the reference for the study that you discussed in your opening slide.
Let's see. Anything I have, you're welcome to. Let's make sure we're on the same page. So what I'm going to do is go to the opening slide. Let's see here. This one about the Cargill stuff, or was it below that? So there's two places where I referenced articles. Number one is the newspaper stuff. Okay, so this is the California, the Cargill, the drought. The other one was the scientific papers referencing production in North American rangelands. These three, I think.
Okay, we'll get it sorted out. Thank you, Matt.
You betcha. And I'll be happy to get whatever you need.
Okay. All right. I think we'll hold any more questions till the end. Matt, will you be staying on the line?
I will.
Okay. All right. Peter Tomlinson from Kansas State will introduce our second speaker today.
Good afternoon. My name is Peter Tomlinson. I'm at Kansas State University, and it is my pleasure to introduce Dr. Megan Rolf with Oklahoma State University's Department of Animal Science. Megan is an extension specialist and researcher at Oklahoma State, working in the area of genetic improvements of beef cattle, and originally hails from Kansas, completed her undergraduate work at Kansas State, and then went on to the University of Missouri for her master's and PhD. And with that, I'll go ahead and turn the floor over to Megan. Are you sharing? And Megan's slides are sharing, so you are ready to go, Megan.
All right, perfect. Can everybody hear me?
I've got you loud and clear.
Okay, perfect. Well, first of all, I just wanted to take a minute to thank you guys for having me here to talk about our project. And our project is related to beef cattle selection and management for adaptation to drought. So when I typically think about drought, in the past, really, my thoughts focused on diminished feed resources and decreased availability of feed and potential for overgrazing if we don't cull back to help preserve those forage resources. But with the last big drought that we had in 2012 in this part of the country, really started thinking about how drought impacts water resources and how that really impacts cattle, particularly out on pasture where they have ponds as water resources. So as I started digging into water, you know, I really think that water is something that we often forget. You know, we balance rations for nutrient composition, and we really don't think about water, but it's an often forgotten essential nutrient that does play a really critical role in every type of life process, anything from transporting nutrients between cells, metabolic processes, regulation of body temperature, which of course plays a role in heat stress, digestion, absorption, and utilization of all the other nutrients. And of course, we all know that water is the most abundant component in living organisms. And research pretty clearly shows that there's a positive relationship between access to clean drinking water and performance factors such as growth, reproduction, and milk production. And I don't think that's going to be a surprise to anybody. But as you sort of think about water use in the beef industry and the water that we use on a farm or a ranch that cattle drink, I hate to say this, but it's really not important on a life cycle assessment scale. So by a life cycle assessment scale, I mean taking into account everything, every input from the tractors that go to plant the crops in the field, to the rainfall that falls on a pasture that the cattle eat, to consumers, retail and consumers on the other end. And if you take a look at where water really is used in the beef industry, it's really overwhelmingly in the irrigation of pre-harvest water and feed, which I think really underscores the importance of feed efficiency in the beef industry to really preserve water resources from that side of the scale. But I think if you think about existing water resources for an individual farmer or rancher, it's pretty easy to see that conservation of those resources is going to be really critical to providing that access to water that those animals are going to need. So if we think about how drought impacts water resources on a single farm or ranch, Of course, you have the problem of decreased water availability. And if we think about a classic summer drought where our ponds start to dry up, this is sort of amplified by the fact that evaporation can cause those resources to sort of diminish as well. We can have decreased water quality and an increase in adulterants in those water sources, and that can affect taste and odor, which of course impacts palatability and whether those animals want to actually drink that water or not. And there's a paper in the literature that suggests that in their study they saw decreased performance, which was likely due to the fact that those cattle just didn't want to drink the water that they had available to them. So they had reduced water and, of course, reduced feed intakes when we have reduced water intakes. We can also see increased water temperature, which goes back and forth in the literature a little bit, but typically that will increase water demand in those animals and increase their water requirement. And this is particularly apparent in taurine cattle. So taurine cattle are those that would be typically non-tropically adapted, so more temperate adapted cattle. And taurine cattle make up the majority of the breed composition of cattle in the U.S. So this sort of sets up a perfect storm scenario in my mind where these animals need to consume a little bit more water, particularly when we think about regulating body temperature as well. When we have less water available, and they may be less likely to want to drink it if we have odor or palatability issues. So this can cause sort of a voluntary water restriction in those animals. And really the concern here is that it can increase the spread of maximum and minimum body temperatures, and as a result of that, really interfere with body temperature regulation and exacerbate problems related to heat stress. And like I like to say, if you're not in drought right now, it's easy to kind of think that everything's okay and that we don't need to be planning for it. But I'm going to argue if you're not in drought, you probably will be in drought at some point in time. So it does pay to kind of think about some of these things in advance and how we might address them, even if times are relatively good at the moment in a lot of the country. So if we take a look at the scientific literature related to beef cattle water intake, sort of the gross estimate is that beef cattle consume 760 billion liters of water per year. And of course, as you might imagine, that's influenced by a lot of different parameters, both related to the animal and to the environment, including the size or weight of that animal, the dry matter intake of that animal, and things in the environment such as the ambient temperature and solar radiation. So there was a really nice study done in 2012 that looked at individual water intake estimates using environmental data. So they had 8,000 pen records from a feedlot in Colorado, and they took a look at trying to estimate that individual water intake. And the R-squared that they found was relatively low, 0.32. But it did underscore that there's probably significant individual animal variation that underlies water intake in these cattle. So if we start taking a look at data that's out there on individual beef cattle, there's some old work in the 50s that shows that Brahman cattle drink less than Hereford cattle at similar temperatures. So Brahman would be a Bos indicus tropically adapted breed, and Hereford would be Bos taurus temperate adapted breed. And these animals in this study were housed individually and fed and watered individually. So we've seen in feed intake the data that can impact feed intake, so it may be impacting the water intake as well. But I think it's a great study to sort of underline some of the differences that we have between breed types and adaptation statuses in these animals. So this study here looked at daily water intake at a variety of levels of temperature humidity index. So this was also pen water intake data, and then they extrapolated that back to individual animals. But they found some differences between summer and winter averages, which you would expect. So their overall average is just a little bit less than 25. Summer was higher at about 32.5 and winter lower at about 17. And a lot of the things that I've showed you other than that study where they were individually housed were studies where they'd collected data on pens of animals. But we really now have the technology to collect large numbers of water intake phenotypes on individual animals where we're still housing them in sort of a normal pen environment, which I think is a great advancement. So two of the systems that can do that are the Grazing system and the Incentech system. And as you can see from these two studies here, now this is dairy, not beef, but there's a really large range in water intakes that you see on these individual animals on a single day. So if we look at the only beef study I can find in the literature related to some of these new technological systems to collect this data, study by Brewer and others in 2011 looked at water intake on 146 growing cattle. They were at thermoneutral conditions, so shouldn't have any issues with heat stress or cold stress here. And they found the mean water intake was about 30 liters per head per day, so not too far off from kind of smack in the middle of the estimate from the pen intake data between summer and winter. They also found a difference between temperate adapted and tropically adapted cattle. So they found that Brahman and Romo Sinuano cattle drank less than British and Continental cattle. So the British and Continental would be those temperate adapted, and the Brahman would be a Bos Indicus breed. And Romo Sinuano is a little unique in that it's a taurine breed, just like those British and Continental cattle, but it's a tropically adapted taurine breed. So we see that those animals that are tropically adapted drink less than other types of cattle. They didn't find any difference between bulls, steers, and heifers in this study, but that may be because they were kind of in a growing phase where we wouldn't necessarily expect large differences in size. So the question in my mind as a geneticist is really, is this a heritable trait and can we make progress from a selection perspective? We really don't know the answer to that in beef cattle, and that's one of the things that the study I'm going to tell you about is really endeavoring to answer. But it is a heritable trait in mice, so there are studies out there that have shown you can select for either a lot of thirst or very little thirst in mice. And they also have a study that estimated the heritability of water intake to be about 0.68, which is pretty darn highly heritable. And they estimated that using 28 inbred strains of mice over a 4-day period. And I think the most interesting thing in that study for me was that there is a large amount of variation in those strains of mice. And there is at least a 2-fold difference between the lowest and the highest strains. And there was a lot of variation that was independent of body weight. So if you take a look, here are the water intakes on the left that are adjusted for body size, and then the raw intakes are on the right side of the screen there. But you can see there's a lot of different variation between those different mice strains in terms of how much water they are drinking in a day. So if we take a look at the relationship between water intake and feed intake in those mice, again, you can see a lot of variation in those different strains. So because of that, we really wanted to ask the question if we could select for increased water use efficiency in cattle to conserve water resources on the farm or ranch. And I think this can really be applicable before in terms of drought planning and also during drought to kind of conserve those resources. So we were fortunate enough to obtain some funding from USDA. It's a 5-year integrated project with research and extension components. And the goal is really to look at adaptability to abiotic stresses. So we're looking at water availability and temperature-related stresses, and we have research and extension components. And our overall goal is to develop beef cattle selection and management tools that really address conservation of water resources on the farm and adaptation to climate variability, and in this case, really related to drought. So to accomplish these objectives, we've been working on a study in the Incentech system, and if you'll recall, I mentioned that this is one of those types of systems that lets us collect individual animal water intake data. So I'm going to show less results than that did, unfortunately, fewer results than that did, because we're not even quite 2 years into this 5-year project, but I do want to show you a little bit of preliminary data. So let me tell you kind of how the study is set up. So got this Incentech system, and you can see these are actually animals that were enrolled in the trials. And you can see the feed bunks most clearly, but they operate all the same way. You've got these bins, and they're on load cells. And if you look at right over here on the far left-hand side of the screen, you can kind of see a gate right here. And that's how these systems work. when they come in and approach that system, it reads their tag, records their individual electronic identification number, and it will drop that gate and take the beginning weight, and then it will let them into the system. They can eat or drink. The water bunks operate the same way on the load cells. And then when they're done, they're back out of the system. The gate goes up, and it takes a final weight. And subtracting those two numbers, we can figure out what the intake was. And as you can imagine with those gates, it runs on a pneumatic system, so it's fairly loud. We have to spend about 21 days acclimating them to that system and letting them learn how to use it. From there, we do a 70-day baseline test where we try and establish baseline water and feed intake values and also their average daily gain and performance during that time period. And then we actually do a 70-day adaptation test where we're looking at some of those stressors that I mentioned. I'm not going to talk about that today just because we don't have enough time to get through all of that. So I'm going to focus more on the baseline test period. So during that time, we've got 120 animals in the barn at any given time, split into 4 pens, and our goal is to have 840 records when we're done. We're looking at water intake, feed intake. We take weights every 2 weeks so we can look at average daily gain. We also collect a lot of behavioral data, so we have pedometers on a subset of the animals in the pen that'll tell us how much time they spend lying down, how much time they spend walking around, and how many steps they take in that time period, so we can look at movement and differences in activity levels. We also have some temperament measures, shoot scores, and exit velocities. And we have a lot of video data so that we can look at time they spend in the shade and out of the shade because they do have access to shade in these pens. And we can look at how their social interactions between animals, so either aggression or sort of positive social interactions between animals. We have a lot of data on health status, so of course we have the treatment data for health interventions that might be needed at any given time, but we also collect information on hematocrit, electrolyte balance, and blood cell counts in those animals. We collect heat and cold stress data, respiration rates, panting data. And we have a subset of animals in a couple of different groups, Group 1 and the upcoming group that's just getting ready to go into the barn. You're going to have some of the animals with rumen boluses so we can monitor core body temperature. And we have rumen and fecal samples on all of these animals to look at the microbial populations in the gut and try and tie that with performance and see if there are any of those microbial populations that seem to explain differences in water intake. So I wanted to share just a little bit of preliminary results. So this is from 2 sets of cattle that we fed over 2 different summers. So they're all summer cattle. And I basically just split them into thirds here. So there's low water intake animals, which are just the bottom third, the medium are in the middle third, and the high are those in the highest third of intake. And so the mean water intake values are as a percentage of their mid-test body weight. So the low group is just under 8% of their body weight, and the high group is averaging around 12% of their body weight. So the overall average is just under 10%, and that works out to be about 38.3 kilograms of water per day as an overall average. That's a little bit higher than what we saw in the pen intake study from Terry Mader, and a little bit higher than the study in the growth state system at 30 kilograms per day, or 30 liters. But particularly with that brew study, that was at thermoneutral conditions, and we definitely had heat stress events for both of these groups of summer animals. Feed intake ranged from about 2.5% to 3% of body weight per day. So if you look at this graphically for individual animals, you can see there is a fairly strong relationship between feed intake and water intake, and that's what we would expect to see. But I think there are some interesting comparisons here, like these 2 I've highlighted, where they're eating about the same amount of feed on an as-fed basis per day, but drinking dramatically different amounts of water. So I think it'll be really interesting to go through and look at the heritability of this and start to sort of tease out some of the genetic differences between these animals. So we have 4 more rounds of calves to feed yet. Group 4 is finishing up now. we have DNA samples on all of these, and our first 3 groups of animals are getting ready to be sent off to be genotyped in the next couple of weeks. And they will be genotyped on a relatively high-density genotyping platform that'll give us information on 80,000 SNPs in the genome. And the idea is that with that data, we can go through and do a variety of things. One is to estimate heritability for these traits. We can also look at genome-wide association analyses and try to start delving into what regions of the genome impact water intake and water efficiency, as well as adaptability to stress and anything that we've really collected phenotypes on in this study. And the last thing is that we can use that genotyping data to go back and estimate breed composition on these animals. Because we don't have known backgrounds on all of these steers. So we really need to go back and look at what their breed composition is when we do these analyses. I mentioned rumen and fecal samples, and we're going to do some metagenomic sequencing on this project. And the idea here is really to take a look at the tails of the distribution. We don't have enough funding to sequence everybody, of course, because that's very expensive, but we can go back and pull out those with really low intakes or really high intakes, or those that are very adaptable to stressors or not very adaptable to stressors, and ask the question if the rumen, or if there are characteristics of the microbial populations in the rumen or in the intestinal tract in the gut that are relevant to either adaptability or water intake. Unfortunately, we don't have facilities to measure greenhouse gas predictions directly, but we are going to use some modeling to do greenhouse gas predictions on these animals. And it's really a separate project, but we've also been collecting tissues at harvest on these animals so that we can go back and do some gene expression work after the fact to look at maybe differences in gene expression that could be tied to differences in water intake and efficiency. So I want to tell you a little bit about some of the extension aspects of this proposal, because one of them I'm really super excited about. But we really did try to focus on sort of all aspects of sustainability in some way for our extension component of this project. And we chose to define that as long-term business viability, stewardship of natural resources, and responsibility to community, family, and animals. So on the environmental side, we're working with Oklahoma Mesonet to develop a water demand tool that will help producers to manage on-farm natural resources during drought. On the social side of sustainability, we are going to do some work with surveys where we're going to assess consumer attitudes towards sustainability and natural resource usage in the beef industry. And another project that I think addresses the economic side is the Cattle Comfort Advisor, which we're also working with the Oklahoma Mesonet to develop. And the idea is there that we can use decision support tools like the Cattle Comfort Advisor to sort of reduce economic losses, because we can plan ahead for times when we might have heat stress and make management interventions ahead of time. And I think that also addresses some aspects of the social side of sustainability, really, in terms of animal well-being. So I know I mentioned those things, but I wanted to show you an example of the Cattle Comfort Advisor. We're going to have a website with some educational modules that are aimed at both producers and consumers. And I wish we had the webpage up and the address to give you today, but that's something we're going to be working on this summer. So address to be determined on that. But we will also host the water demand tool when that gets developed in the later years of the project, as well as the extension of the Cattle Comfort Advisor, because we hope to have the first test maps in June of this year. So the Cattle Comfort Advisor is a product that was developed by the Oklahoma Mesonet, and it's an Oklahoma-specific product at the moment. But what I really like about it is it tries to relate kind of what the animals are experiencing to a value that sort of makes sense to us as humans, because cattle experience heat stress differently than we do and at different temperatures and different conditions. So what we're working on with Oklahoma Mesonet is taking their Oklahoma-specific tool and using National Weather Service data to sort of expand that to the entire nation. So one of the ways we're sort of calibrating that equation in different parts of the country is using rumen temperature boluses to look at core body temperature in different regions of the country. So if you have a population of cattle sitting around and you have a burning desire to put some boluses in them, and work with us on collecting some of that data, be sure and drop me an email. So I want to take just a few seconds as I wrap up here to recognize the people that are part of this project, not only collaborators at universities, but also our veterinarians, because they've been very essential in this project, and of course Al Sutherland and the entire team at the Oklahoma Mesonet. As I mentioned, we are funded through the USDA, and we're very grateful for that. We've also received some supplementary funding from the Texas Cattle Feeders Association to do some water quality monitoring inside the barn there. And of course, those of you that have been graduate students or been around graduate students know that on these big projects, the grad students are doing a lot of the day-to-day animal care and data collection, so I wanted to take a second to recognize the graduate students and undergraduate students that have contributed to this project. And with that, I'd be happy to take any questions that anybody might have.
All right, Megan. Well, thank you for a very interesting presentation. We do have a poll question up that we would appreciate everybody answering. And while you're doing that, I will monitor the chat and the Q&A for any questions for Megan. All right, we do have a couple that are in the Q&A section. So
Okay.
Has anybody looked at epigenetics of if water stressing during gestation and early growth has long-term impacts on water intake?
That's a fantastic question, and it's something I've wondered as well. I haven't seen that in the literature, but I think that's something that would be a great thing to do and would certainly be very interesting.
Okay. We have another question. Is water intake idiosyncratic for the upper tail of the data?
So are we getting at variability here for those animals that are very high on the scale?
I'll ask Mark to provide some more clarification on his question.
So I will say in the meantime, we have found that some animals seem to be pretty consistent in terms of their water intake, and others seem to be all over the scale. And I'm really curious when we start digging into the genetics if there's a genetic component to that, or if it's more behavioral in terms of you know, maybe we have some dominant animals in the pen and they hang around the water trough and drink the water because if they're there drinking the water, they make sure nobody else can. So I think that might be something interesting to look at in our video data. And just out there observing the animals, there are definitely animals that kind of hang out by the water trough and block other animals from accessing it from time to time.
And so, you know, Mark has provided a couple clarifications. One is, do they play in the water? Question mark. And then the comment, many penned animals display odd behaviors.
Yeah, so they do play in the water. I've never been around cattle anywhere that didn't play in the water. The one thing about the design of the water bunks that helps with kind of getting erroneous water splashing data is that the water is actually in the very bottom of those bunks, and there's a plastic insert around, so they actually have a fairly small area to drink out of. So they can splash it out, but it's going to fall into the bunk, so it's not going to be counted as weight leaving the bunk and get erroneously counted as intake. It's going to remain sort of on those plastic inserts in the bunk.
Okay. Great. Do we have any other questions for either Megan or Matt?
Peter, this is Laura. Since we have 1 minute here, I have a question for Matt,
Okay.
if he's back. He looked at changing productivity, but did he look at changing in timing of peak production in forage or grassland?
No, we have a separate study that is doing that, looking at phenological changes and when the maximum, for example, might be expressed, but that wasn't included here yet.
All right. Thanks, Matt.
All right. Well, we'll go ahead and thank both of our speakers for their time today and presenting some very interesting data that hopefully has got all of us thinking as we look to the future. Be on the lookout for our next program in May. We are finalizing the speakers for that right now, and we'll be communicating that information in the near future. And with that, I'll take a final call for any last questions.
Peter, I see there's a question about archiving of the webinars, and I just put that website in the chat box,
Okay.
climateagwater.wordpress.com.
How much benefit would there be for summer-fed cattle to be red-hided than black-hided?
Okay, so I just saw that question myself. Thanks for pointing that one out. So there are differences in heat stress due to hide color, and also skin color can play a role in that. So the black animals or any kind of dark-hided animals and dark-skinned animals are going to be more stressed as a general rule when you encounter heat stress. In terms of what we've looked at for the water data, we've got coat color recorded, and that's something we're definitely going to look at. We haven't run that analysis yet, but some of that may be mitigated a little bit in our study because they do have access to shade.
All right. Well, I'm not seeing any other questions in the chat box. So again, I thank both Megan and Matt for their time today. And Laura or Hans, do you have any closing comments?
No, thanks, Peter.
Okay. With that, we'll conclude today's webinar and look forward to seeing you all next month.